This session will be moderated by Dr. Valish. Dr. Valish is a senior consultant in medical writing and biostatistics with Marksman Healthcare Communication. He completed his MD Pharmacology from Grand Government Medical College and JJ Group of Hospital, Mumbai, and MBBS from Kempegowda Institute of Medical Sciences, Bangalore. He has 9 years and 6 months of experience in teaching medical pharmacology to various courses like MBBS, MD, BDS, BSc Nursing, and Allied Health Sciences. He is a member of various organizations like International Society of Pharmacoeconomics and Outcome Research, Indian Pharmacological Society, Association of Physiologists and Pharmacologists of India. He also served as Basic Medical Scientist at Velamal Medical College, Sharavana Hospital Ethics Committee. The session will also be chaired by Dr. Sangeeta Raja. Madam is an Associate Professor from Department of Pharmacology from SRM Medical College Hospital and Research Center. She is also a faculty from SRM Center for Clinical Trial and Research. She completed her MBBS and MD Pharmacology, post-graduation from SRM Medical College. She has 7 years of teaching and research experience. She had received gold medal for securing university rank in MD Pharmacology, and she has been consistently involved in various clinical trials at SRM Hospital. I invite Dr. Sangeeta Raja to introduce the speaker.
Yeah. Thank you, Dr. Indumati. Am I audible?
Yes, ma'am.
Yeah. I would like to thank Dr. Melvin George for giving me this opportunity to chair the session. And it is my pleasure to invite Dr. Maulik Doshi and to introduce him. Dr. Doshi did his MBBS from BJ Medical College and also his MD from BJ Medical College, Ahmedabad. And he did his DM in clinical pharmacology from Seth GS Medical College, Mumbai. He has got a 10+ years experience in diverse areas such as regulatory presentation, medical writing, medical monitoring, medical data reviewer, clinical operations, pharmacovigilance, medical affairs, statistics and data management in various therapeutic areas like cardiovascular, endocrinology, dermatology, ophthalmology, to name a few. The list is huge. He has successfully completed end-to-end clinical trials and received approval for many products. And he has got a close rapport with KOLs and subject experts in various therapeutic areas and contributed to more than 80+ regulatory studies in different phases of drug development. He also has numerous paper and a book chapter he has authored. And he also has some educational activities on social platforms like an education forum on Facebook named Clinical Pharmacology with over 14,000 members from all over the world. And he's also running an educational channel on YouTube. And he seems to be multilingual and he has got a diploma in French, which is fascinating. And it's a pleasure to invite you, sir. The floor is yours, Dr. Doshi.
Yeah, thank you very much. Am I audible?
Yes, sir.
Dr. Valish, are you there? Yeah, I'm here. Good day to all the speakers and participants. At the outset, I thank the organizers for giving me this opportunity to share this very interesting session that deals with Indian regulations surrounding biosimilars. I mean, this session is extremely interesting because, as we all know, India is a pharmacy hub of the world where manufactures and exports most of generics. But what about biosimilars? To my knowledge, the biosimilar market has not yet been dominated by India, and that day is yet to come. And some reasons for this non-dominance yet, according to my knowledge, is the laxity of Indian regulations. They have been pointing out that the biosimilars that are approved in India are backed by the lax regulations. So it is really interesting to know what are all the regulations with respect to biosimilars in India. They are not called biosimilars as such; they are called as similar biologics. So, are there any other differences? What exactly are these differences? The latest regulations came out in 2016. So, what do these guidelines say? How do the Indian regulatory processes compare with those of the Western world? So we are curious to know about this. So no better person than Dr. Maulik to introduce these topics to us and share all our, clear all our doubts. So I invite Dr. Maulik to take over the floor from now.
Yeah, thank you, Dr. Valis. Is my slide visible to all?
Yes, sir.
Okay. So first of all, I would like to thank the organizer and the Dr. Melvin for establishing this wonderful workshop. Because nowadays biosimilars are gaining so much of traction all over the world, and it is very important that the young pharmacologist, young researcher, they are getting exposure regarding the various regulations and whatever studies are required, so that it will help them whenever they are associating with this biosimilar journey anywhere in their career. Okay, so now as a part of biosimilar journey, one is the Indian regulation. Now, this is my standard disclaimer. My outline is I will be discussing on some definitions, not all the definitions, but the key definition. Then the difference between a regular pathway and the biosimilar pathway. Then I will be also presenting the analysis of approved biosimilar and the learning from it. Then as per Indian regulation, what are the analytical characterizations are required? Then what are the clinical pharmacology studies are required? The importance of clinical pharmacology studies. Then the various elements of these studies like sample size, design, study population, duration, dose, I'll be also covering in this studies. As well in confirmatory clinical studies, same thing, design, sample size, duration, endpoint, I'll be covering. Then the phase 4 plan as per Indian regulation. And as per Indian regulation, what are the criteria required to get fulfilled for extrapolation of any indication? All right. So Dr. Velish asked a very important question that here in India, it is known as a similar biologic. And what we have, the word is biosimilar. So the Indian regulatory guideline mentioned that a similar biologic product is that which is similar in terms of quality, safety, efficacy, and efficacy to an approved reference biologic product based on comparability. So here the important one is similar. And which are the domains? The domain is quality domain, safety, and efficacy. Then what is a reference biologic? A reference biologic as per Indian guideline, it's a comparator for comparability studies with a similar biologic in order to show similarity in terms of safety, efficacy, and the quality. And it is licensed or approved in India for any ICH countries, and it should be the innovator's product. An important point is it should be licensed based on a full safety, efficacy, and the quality data. Now, the earlier speaker, they have already discussed the regular pathway and biosimilar pathway, but I would like to discuss once again because it is having a relevance with my subsequent slides. So now in regular pathway, it's a stepwise approach. So in the first step, analytical characterization is performed, where the primary structure, secondary structure, tertiary structure of molecule is studied, then glycosylation, post-translation modification are studied. After that, the non-clinical studies are performed to understand safety, immunogenicity, and other PKPD aspect. After that, the molecules come into the clinical pharmacology study. In clinical pharmacology study, it is a PK, PD, and immunogenicity. And then last phase, clinical studies, where there will be phase 2 study followed by phase 3 study. Now in phase 2 study, it will be a dose ranging study. So there will be more than one doses will be studied, and based on that, a proper dose will be selected for phase 3. And then the phase 3 program. Now, when we look at a biosimilar regulatory pathway, so what you can notice the similarity is it is also a stepwise approach. Like you do characterization first, non-clinical followed by clinical pharmacology, and the comparative clinical study. But here, what you can notice is the amount of data which is generated at each domain. Now, when you notice this regular pathway, so amount of data is higher in clinical studies than the first step, that is a characterization. Okay, but in biosimilar regulatory pathway, the amount of data, so when I say amount of data, it means the data which can show, which can quantify the similarity or any differences with the test product and the reference biology. So the amount of data which is generated is the highest at the first stage. And then the next step of studies, it depends on this step. So if there are any differences reported during this analytical characterization phase, then only studies related with those differences will be done in the non-clinical study, or some routine studies will be done in the non-clinical study. Then in clinical pharmacology and the comparative clinical study. So here, the important point which I want you all to know is that in biosimilar pathway, the initial phase is very much important, very much vital to establish the biosimilarity. Now, the key questions, like which are the domain for which data is generated for biosimilarity? Is it mandatory to show biosimilarity in each domain? What if a product doesn't show biosimilarity in one domain? Will it still get approval? And which are the critical domains in which product must show the biosimilarity? So I am presenting analysis of biosimilar studies. Okay, so here what you can notice is a domain that the quality, functional, PK, PD, clinical studies, that efficacy and safety study, then immunogenicity study, and the result of that for the product. Okay, so let's understand the first biosimilar. Example is the trastuzumab. So this trastuzumab, it showed its similarity with the reference Herceptin in quality, functional, and PK. Now PD is not possible, but when they did a clinical study, so clinical study did not go as it was planned. It failed to meet upper equivalence limit. So you have a upper and lower limit, and you have to follow all the limit like 80 to 125, but it actually failed to meet this upper limit. And in immunogenicity, it showed the similarity with the reference product, and the product got approval. Same is the case with trastuzumab. It fulfilled the first three domain, quality, function, and PK, but the same result with the clinical program, like it failed to meet upper equivalence limit. Immunogenicity was fine, and it was approved. Then rituximab. Rituximab showed similarity with quality, function, and PK, and then the study was proceeded to the next phase. But in clinical study, it met the primary endpoint, but it didn't show some uncertainty. It showed some uncertainty for progression-free survival and overall survival. So it did not go as per plan, but still product got approval. Same is the case with the rituximab. Then the next product is the teriparatide. Okay, and for teriparatide, PK, it showed the 95% confidence interval. It showed it was within the limit, but the unity was not in the 95% CI. And PD showed fine, and clinical study was not applicable, and the product was approved. Then rituximab, it did not show a good similarity. Some only like decreased ADCC activity was seen. Otherwise, everything was fine. PK was fine, and clinical and immunogenicity was fine, and the product was approved. Then the last drug, pegfilgrastim. The pegfilgrastim, it showed a similarity with quality and functional, but it actually failed in PK, and rest all are fine, and the product was withdrawn. So here what you can see that when a product shows a similarity with quality, functional, and PK, then it makes sense for a sponsor to proceed with the next phase. If it doesn't show, then sponsor will not go to the next phase. Okay, and it is not the only one domain or two domain are being important. It is a totality of evidence approach. So all the domains are studied. The data is reviewed for all the domain, and based on then a decision is taken. And the third point is for a biosimilarity, quality, functional, and PK. these are very much important. If there is any issue with this 3 domain, then there are chances that either sponsor will not proceed with the next phases, or the regulator might not give approval for the product. So now we'll understand now the step 1, the analytical characterization. So in analytical characterization, the discussion will be on structural analysis and the functional analysis. So as per Indian regulatory, 4 things are being studied under the analytical characterization: structural and the physicochemical property, biological activity of the product, so that depends on the mechanism of action, immunological property, whether the product is immunogenic, and if it is immunogenic, then is it producing a neutralizing antibody which can impact PK or PD of the product. Then purity and impurity. So basically, the idea of doing this extensive study is like to quantify the similarity and to quantify the differences. Okay, so for example, a product which is being tested against the reference biologic, and these studies are done, the results seem that there is a similarity in primary, secondary, and tertiary structure. There is a similarity with the biological activity and immunological activity. However, there is no similarity with glycosylation, phosphorylation, and impurity. Suppose this is the case, then what to do? Then what scientists will do, scientists will understand what will be the impact of these differences. So this is called as a residual uncertainty. Okay, so the further test will be conducted by the scientists to understand these differences. And based on that, the future studies will be conducted either in animal or in clinical studies. So as I said earlier, in a biosimilar program, this first step is extremely important. Then we'll talk about the preclinical study. In preclinical study, there will be a toxicity study, PK/PD, and immunogenicity. Now, as per Indian regulation, there are some regulatory bodies which come into the place, some committee come into the place for getting approval of this product, and then also review of the result. So a proposal is prepared and it is submitted to the Institutional Biosafety Committee, which is known as IBSC. Then this application with some information like proposed use of biosimilar project, biosimilar drug, what is the target population we are planning, what are the diluents, whether the product is in cartridge or vial, the route of administration, dosage and mechanism of action. Then the data of reference biology. The similarities, route of administration, dosage and mechanism of action. But the thing is, like, the reference biologic is approved in how many indication, which are these indication, what is the efficacy in each of these indication, the peak and tox data. So all this basic information is compiled and it is submitted along with preclinical protocol plus the IBSC approval to RCGM, which is called as Review Committee on Genetic Manipulation. Now this RCGM will review the protocol and then they will give approval for a preclinical study. Then preclinical study, pharmacodynamic study, and toxicity studies are conducted. Then again, a sponsor will go to the same committee with a review of those data. So RCGM committee will review the data, and if it finds everything absolutely fine, then they will give approval for human clinical trial. And then after that, the role of TCGI will come. So now let's understand what are these tox studies, which toxicity studies are required, which is not required, or which are optional in my subsequent slide. So in toxicology study, repeated toxicity is mandatory. Okay, and the other studies like safety pharmacology, then reproductive toxicology study, mutagenicity and carcinogenicity, the requirement of this study, it will depend on the data of toxicity study and the analytical characterization. Because what we have seen in the earlier slide, that there was no similarity with glycosylation and phosphorylation. So whether because of that, is any of these toxicity studies is required to conduct or not. Okay, so based on the analytical data, the next plan will be prepared. But irrespective of the analytical characterization data, repeated dose toxicity is required. Now, if this repeated dose toxicity is conducted, then how it should be conducted? So the first point is choice of animal model. So the animal model should be selected which is relevant with the disease condition. Okay, and if the animal model is not found as per disease condition, then any rodent or non-rodent molecule can be taken. Now, when the animal model is taken, the route of administration, it should be the intended route of administration. Then the toxicity study duration, it should be of at least 28 days with 14 days of recovery. And the dose which is studied in this toxicology study, so it will be against a vehicle. So one arm will be vehicle, and then there will be 3 or 4 arm of the dose, different dose level. So 1x, 2x, and 5x of the human equivalence dose will be tested in animal, and then the study will be conducted. Data will be analyzed, histopathological and all the data will be analyzed, and then it will be presented to the RCGM. Now, pharmacodynamic study. Pharmacodynamic study, it can be conducted in vitro and in vivo. So in vivo is important when in vitro is not possible, but when you conduct in vitro, it can be like cell proliferation study, cytotoxicity study, then a neutralizing study, and the receptor binding assay. These studies are conducted, and once the sponsor conduct all these study, it gradually try to minimize the residual uncertainty about its test product. Then the next step come is the clinical study. Now in clinical study, there will be clinical pharmacology study in which PK, PD, and immunogenicity study will be conducted. So some quick question now again. Why clinical pharmacology study, which is PK, PD, are important in biosimilarity program? When one should select healthy or patient population, when one should select crossover design or parallel design. If study is done in healthy, then should the dose be therapeutic or it can be lowered to maximize safety? How to select the margin, equivalence margin? How to select pharmacodynamic endpoint? Can we combine clinical pharmacology study with comparative clinical trials or not? So I would like to address this question in my next slide. So first is the importance. Clinical pharmacology study, it is next to address a residual uncertainty after analytical characterization. It adds a very critical evidence of biosimilarity in terms of exposure, which is by PK, then exposure and response, which is PD. The most important part is that when a sponsor finishes a biosimilar study, sponsor is also interested to extrapolate to other indications. Now, for other indications, sponsor is not going to do a very expensive and long-term phase 3 study. Okay, so when they want to do extrapolation, then the clinical pharmacology data, it supports in those extrapolation. So that is the importance of a clinical pharmacology studies. Now let's understand the different kind of nuances in this clinical pharmacology study. What should be the studies design? So the principle is the study design should be sensitive enough to differentiate any clinically meaningful difference. And it should be also done in the lesser sample size. Okay. So now we all know there are two options available. One is a crossover option, other is parallel option. Now the crossover option is very sensitive. Okay, because one subject will act his or her own comparator. Okay, so the variability is very much lesser in crossover design. Plus crossover is conducted in healthy, so you don't have those disease burden kind of issues and variability in healthy. So it is considered the most sensitive design and preferred. Because there is a less variability, which is a coefficient of variant, and because of that, you will have lesser sample size. Then if the study is going to conduct in healthy, then crossover is the design of selection. If the product is having shorter half-life and rapid pharmacodynamic response, then the crossover design is preferred. Now, what happened? There are situations that study is not possible to conduct in healthy because, like let's say, for example, some oncology molecule. Okay, In oncology molecule, some molecules are the antibody drug conjugate. So in those things, the conjugate will be a cytotoxic drug. Now you can't give a cytotoxic drug in a healthy. You have to give in patients only. Okay. So if the study is going to do in patient, then it should be done in the parallel design. If the product is having longer half-life, which makes a crossover design with washout very impractical, then a parallel design is preferred. And if there are chances that the repeated dose can result into a different immune response, then also a parallel design is preferred. Then let's come to the study population. The principle is the study population should be safer, sensitive, and it should be also representative of the target population. Okay, so a healthy are selected in PK studies because it is considered more sensitive, lesser subject, the shorter T half, and rapid PD response. And patients are selected when it is not possible to give in healthy because of safety issue, or there is a longer half-life or immune response. Now the dose selection. Which dose will be selected in pharmacokinetic pharmacodynamic study? So the principle is the dose should be the sensitive again to differentiate in clinically meaningful difference. Okay. Now that means what is the definition of sensitive dose? So the sensitive dose is dose which comes into the step part of the dose response curve. Now, if the study is going to conduct in patient, then there are two options for the dose. A sponsor can take approved dose. Like suppose the study is conducted for trastuzumab, then the dose of trastuzumab 3 mg per kg can be taken for patient study. Now there are instances where if this approved dose, when given, if it causes non-linear PK, or if the dose exceeds maximum PD effect, in those conditions, it is not advisable to go with the approved dose. So for those indication, for those drug, lower than approved dose plus in the part of the dose response curve should be selected. Healthy. In healthy, the lower dose in part of step part of dose response could be selected. So there is a maximization of the safety over the risk. Route of administration. What should be the route of administration in PK/PD study? It should be the same route of administration as reference. Now there are chances that reference will have two route of administration, subcutaneous or IV. So in those two route, one should select the subcutaneous route. Okay, because this is the route where there are chances of variability. So it should be selected subcutaneous. Statistical design. It should be equivalence design. So the usual the approach is to take 80 to 125 approach. However, if the sponsor can justify other design like symmetrical or asymmetrical margin based on the dose related toxicity, then it is also fine. Now let's discuss that whenever the pharmacodynamic studies are conducted, so what should be the pharmacodynamic marker and what should be the characteristic? So the pharmacodynamic marker, whatever is selected, a sponsor has to justify that it fulfills 5 criteria. The first criteria is mechanism of action. The relevance of PD biomarker to the mechanism of action. And second thing is there should be the data on the onset, time of onset of the change with the PD marker related to the dosing. So one criteria and two criteria. Now let's discuss these two criteria here in a real example. So here, this is apoetin alpha. Okay, so this acts on this erythroid progenitor cell, and then erythropoiesis begin, which cause increase in the reticulocyte count, followed by change in the hemoglobin, and then there's the clinical outcome. So these two markers, reticulocyte count and hemoglobin, these two markers fulfills this first condition, that they are between the mechanism of action. Second thing is time of onset. So this picture, it gives a idea about the time of onset. This reticulocyte count is time of onset is early onset, and this hemoglobin is late onset. So these two marker has fulfilled two criteria. It is not only coming in the mechanism of action, also it is possible to characterize the time of onset. Okay. Now, why time of onset is being considered? Because depends on that one can decide the duration of study. Suppose a reticulocyte count is taken as a pharmacodynamic endpoint, then the study will be of short duration. And if this one is selected, hemoglobin, then the study will be of longer duration because it's a late onset. Okay, so this is why the importance of this time of onset is taken. Now let's discuss the two other point of the biomarker. The first is the dynamic range of the pharmacodynamic marker over the exposure range to the biological product, and then the sensitivity of this PD marker to differentiate between the proposed biosimilar product and the reference product. So now let's discuss these 2 points in this example. So what happens that if the PD marker, it should have a different curve according to the different doses. So this characterization should be possible with the marker, and then the dose response relationship is taken, and then it is possible to have this sensitive dose range. Okay, so this is how any pharmacodynamic markers validation is conducted, and if it fulfills all of these 5 criteria, then the pharmacodynamic marker is considered as a part of an endpoint in PD study or in the phase C clinical trial. Now let's discuss on the phase C clinical trial. So some key question again. What should be the design of the phase C program? What should be the objective? Like we have already seen the PK. Is it important? Is it need to again do PK in phase 3 program or not? Then when a reference biologic is approved in more than one indication, then which indication to select for phase 3 study? What should be the trial duration? And what should be the sample size? So the study design, it's just standard. It should be randomized, parallel group, blinded, and comparative study. Now coming to the objective. So objective, of course, it's a phase 3 program. So objective is efficacy and safety. Okay. However, whether to do PK in phase 3 program or not, it depends on the earlier study. So earlier PK study, if it is conducted in healthy, then it is absolutely mandatory to do PK in the phase 3 program. But if the earlier PK is conducted in patient, then some PK time point can be waived off, but not absolutely. Okay. Then immunogenicity as a secondary. Study population. So study population should be a population which is an approved indication. Now here comes some kind of options. Like if you talk about trastuzumab, okay, so trastuzumab, it is approved in 2 indications. One is early breast cancer and other is metastatic breast cancer. So now again, which indication one should select? Should we go with EBC or should we go with MBC? So it depends from this in these 2 indications, which indication is more homogeneous. Why? Because what will happen that tumor burden, then other antibody level, so many things will have a confounding factor. Okay, so if there are as much of lesser compounding factor, it is important and it is also beneficial for sponsor that they will be able to demonstrate the similarity. So the population should be the sensitive enough. So in other words, it should be the homogeneous for the condition. What should be the endpoint? So one can select a reticulocyte count like PD measures, then some surrogate endpoint like objective response rate. Now we all know that if you talk about the breast cancer study, then the interest in the survival, right? Progression-free survival or overall survival. But these survival endpoint, which are hard endpoint, these endpoint are not considered sensitive enough. Plus, it will also increase the duration of study. So that's why it is mentioned. It is recommended by regulatory agency that you select endpoint which are sensitive enough. And these are so in EBC and MBC study, the objective response rate is a sensitive endpoint, and that's why it is selected as a primary endpoint in phase 3 program. Trial duration. The trial duration again depends on which endpoint is going to select. Now sample size. So as per Indian regulation, As per Indian regulation, at least 100 evaluable in test term is mandatory in phase 3 program. Statistical hypothesis. It can be either equivalence design or it can be non-inferiority design. Now, in equivalence design, there are two options. The margin can be symmetrical, 80 to 125, or it can be asymmetrical. So, the asymmetrical margin will give a lesser sample size. So, there is the incentive to go with asymmetrical margin. However, there are some principles when to select which margin. So, if the dose related toxicities are there, then it is advisable to go with the symmetrical margin. And if it is not there, then one can take the higher end of upper margin, asymmetrical margin, and do the study. Now, Indian regulation also says that because we all are saying that in biosimilar program, the critical part is the earlier test like functional, quality, PK, PD. Okay. So does it mean that there are any chances that one can actually waive off the entire phase 3 program if the earlier data is so much robust? So the answer is yes. It is possible to waive off the phase 3 program provided the product meets all these criteria. So quality and functional characterization, there is a high similarity. In preclinical evaluation also there is high similarity. There is a similarity in PK also and PD also. And the firm is ready to do a phase 4 study. Then in those situations, a phase 3 study will be waived off. Now, when the phase 4 studies to be conducted, then the study design, it can be a single arm, non-comparative study. The population should be as per the approved indication. The endpoint is like safety is important first, followed by efficacy and immunogenicity. Sample size, it should be at least 200 evaluable. So 200 in phase 4, 200 in phase 3. The total at least 300 patients are required as a part of a biosimilar package. However, if the condition is rare, then there can be some exception. Now, once a sponsor finishes a phase 3 program and all other data, and then they submit to the regulatory agency, then sponsor also wishes to market that product in other indications. For example, trastuzumab, if sponsor finishes study in EBC, then also they would like to market in MBC also. So they would like to get approval in EBC and MBC both. So for that, what data should be submitted? So the quality and functional data, preclinical data, clinical safety, which is already generated. Now the mechanism of action, it should be same for other indication as well. They have to prove it. Okay. And the involved receptor are same for other indication. They have to prove it. If these 5 conditions are fulfilled, then the DCJ will grant extrapolation to the other indication. So now take home message. So I want you all to go at least with 5 points today, that the clinical development plan is a stepwise approach. The data which is required to be generated is from quality, functional, PK, PD, efficacy, safety, and immunogenicity. Now, after this all, the quality, functional, and PK, PD are very much vital in addressing any residual uncertainty. Need of phase 3 study, it depends upon the status of residual uncertainty. So in most of the case, there will be some residual uncertainty. So phase 3 is there. However, there are chances that if a sponsor can show that all these are fine, like in case of pegfilgrastim, then there is no need to do phase 3 study. And in phase 3 program, the requirement of design and everything is very standard like other program. Only thing that Indian regulation, they are specific on the sample size. So what they are saying, phase 3 should be conducted in 100 patients, and phase 4 to be conducted in at least 200 patients. Yeah, thank you.
Thank you, Dr. Maulik. It was a clear and comprehensive presentation. I have a few questions. I hope you will be able to answer that. You are speaking about the sample size of as per the Indian regulations in different phases. You said 100 sample size in phase 3 and 200. I mean, what is the rationale in fixing the sample size? My understanding is that it would be much better to arrive at a sample size rather than giving a fixed number. is it actually the standard, or what is the logic behind coming up with this fixed number? That is my question.
So this is a very logical question. So what happened, I will tell you the background. When this guideline was prepared, they invited representatives from the industry people. Okay, so everybody give their suggestions. So at that time, what the discussion happened and what I understand is that, okay, you can come to definitely with a sample size proper calculation. So when you go to the DCGI and DCGI SEC meeting, when you present sample size, they will ask, how did you calculate sample size? So anyway, that proper justification of sample size is required in DCGI. But what they are saying that since you are doing only a phase 3 program and you are not generating any patient data, we believe you at least your sample size should be at least 100 in phase 3, and total you at least generate data of 300. Okay, so the sample size has to be calculated, but this is what they say, at least you generate this much of data for Indian population, so that we can assure okay, there are enough data and the product can be continued.
Okay, thank you for the explanation. I had another question. You said that for some, if some criteria are satisfied, entire phase 3 studies can be waived off according to Indian regulations. If a product gets approved because as a result of such a waiver of phase 3 studies, what will be its status when it goes to global markets? I mean, do other regulators as well follow similar patterns of phase 3 waiver of? Do other markets accept such a product that has not undergone phase 3 studies because of provisions in the Indian regulations? This is my question.
Okay, fine. So now let's say that there is an Indian sponsor, and this Indian sponsor has approved is biosimilar based on Indian guideline, and in which they have shown similarity with quality functional PKPD, but they haven't done any phase 3 program, and they got Indian approval. Okay, now if they want to go some other markets, in other market they will follow their guideline. So when you go to other market, there can be two market: regulated market, non-regulated market. Okay, now when we look at regulated market, then they are also of the same opinion that it is not required to do a phase 3 study. It is not mandatory, because what now literature is saying that phase 3 program will not take much value if you have already shown quality functional PK and PD. So that's why it is not mandatory to do phase 3. So their person, the company can get approval in other market also, but they have to do and they have to follow some other requirement for the particular market.
Okay, thank you. I think we have a question from Dr. Srikanth Gattu in the chat box. Can you please comment on the Indian regulatory authorities' strengths and weaknesses in assessing complete data dossier of submission? Areas where they are not strong and depend on other country opinion, and areas where they are strong and are role models for other countries. So this is the question by Dr. Srikanth.
So what we are seeing that over a period of 4 to 5 years, the Indian regulatory are also helping the industry a lot. Okay, they are also following the international standards. So if you see the Indian guideline, and if you see the other guideline also, they are almost same. Now, only thing is, like, if you ask what are the weaknesses of Indian regulatory, then what I have observed that the person who is reviewing the entire dossier, his or her background should be related with pharmacology, so that a person can really understand the data, and then based on that, they can take a decision.
Okay. Dr. Sangeeta, do you have any questions?
Yes, sir. Yes, sir. I do have one question. Dr. Maulik, wonderful presentation. I have a question on, there are issues of limited health insurance coverage for biosimilar when you compare that with an originator. So how the regulatories are going to overcome it? Are they having any plans for it? Because most of the patients couldn't afford it.
So biosimilars are come in a market because the patient cannot afford the originator molecule.
Yes.
Okay, so the biosimilars anyway very like 1/10 of the price of the what original molecules are. Okay, so now when these biosimilars are approved, then some institute put those product in their own list, and they will make them available. So I don't know currently that some insurance program is going or for this Indian biosimilar or not.
Okay. Okay, sir. So there's one more question from Dr. Smita. Do you think Indian biosimilars are doing less testing for the domestic marketing compared to the overseas market?
So, well, they cannot afford doing a less testing, because what will happen that if they do less testing and if product fail, uh, it's a huge loss to the company only. Okay, so only thing is that some company has their some specific requirement. Okay, so maybe domestic requirement and international requirement are different, so one has to fulfill the requirement, but all the basic ground level all domestic company have to do, so that they not only be successful in their phase 3 studies and can also be the competitive when it is in the market.
Okay. Okay, sir. There's one more question from Sujay S. Patil. There's a term called biomimic. So is it semi-regulated countries product developed can be called biomimic? So is it semi-regulated countries product developed can be called biomimic?
So basically there are only two things, whether it is a similar biologic or not, and if the product is fulfilling all the requirement as a similar biologic, then it depends, and then it will be called as a similar biologic. There are no different things, it's a very clear.
Okay. Okay, sir. Is there any other question? Or shall we move on? Okay, if there's no other question, thank you so much, Dr. Bhaumik.
Yes.
And we will move on to the next session.